in the DNA, we have a lot of just maybe you could say randomly evolved sequences that don't really maybe matter, and the model needs to be able to identify what matters from what doesn't matter for a given property, and that's key compared to the other approaches.
Hi everyone, welcome to Let's Talk Research, the official InstaDeep podcast where we dive into cutting-edge AI research and the stories behind it. My name is Bora Gilola, I'm a research scientist here at InstaDeep.
And I'm Massimo Bortone, also a research scientist at InstaDeep. In today's episode, we will be talking about genomics. In particular, we will be looking at the evolution of our Nucleotide Transformer family of models, which we will be referring to as NT throughout the episode.
Exactly, and joining us today is our head of Genomics AI Research, Bernardo Almeida. Welcome, Bernardo.
Thank you, guys. Happy to be here.
So, Bernardo, before we jump into the science, we ask this every guest. How did you get into genomics in the first place, and how did you get to InstaDeep?
Sure, yeah. So that starts a bit, a few years back. I think since the end of my studies that I was kind of fascinated by our bodies, our tissues, our cells work, and mostly by how all this information is encoded in the DNA sequence, so in our genome. And so I started working on that, but at the beginning, I realized how complex this is. And given that, we really need, large-scale data analysis and machine learning to fully understand all this, information and how our DNA encodes us as a human being. And so I joined, research projects on genomics, large-scale data analysis, and in my PhD, I started developing the first models to really try to understand this code. At the time, it was a very specific tasks, specific regions of the genome. But to really make an impact, these models, I think they need to, have a larger scale, be more foundational, similar to what we see in other fields like vision, natural language processing. And that was my entry point to InstaDeep. So really develop models at scale, contribute to the community, models that people can use, and that take a lot of genomes, a lot of data all together into the same model and power various applications in the field. And yes, so now I'm heading the department, but that's really our ambitions in the company.
Okay, so let's take a step back and explain maybe what is DNA, what is the genome, and how is it maybe different from actually modeling language? Because I think a lot of our listeners will be familiar with that, but maybe not with the genomic side of things.
Sure, yeah, so that's key. So if we start very basic, so DNA is a molecule that each of our cells has. But it's a molecule that contains all the information to generate all our different cell types during development, different tissues, organs, etc. So we can think about it as a book, for example, with all the information, all the instructions. And it's the same book inside each of our cells. And that book varies, for example, between different individuals. That's why we are all different between each others, between different species, etc. And so that book is like, in humans, 3 billion letters long, but each letter is just has a vocabulary of 4 different letters. So it looks kind of simple, but then all the complexity emerges from the patterns that are present in this book. So the information is not coded in each nucleotide alone, but really comes from motifs, combinations, patterns that operate at different distances in the sequence. And that's one of the main aspects why this is different from a different sequence level problem and data. So the information is sparse, relies on combinations, and differs, although it's similar, differs between different cell types in how it's used.
So kind of taking this a little bit further, right? So when a cell kind of achieves some sort of function, it does so usually via a protein, right? It's either an enzyme or an antibody or a recognition molecule, right? A receptor, perhaps. So these are all encoded for by individual genes. And you're saying that the kind of whether a cell or a tissue, for example, is going to perform some function by expressing that protein comes down to how it's reading instructions from that book, right? So how is it that kind of the same copy of the blueprint or the book, so to speak, is in each cell, but different cells read different parts of it and behave differently as a result of that?
Yeah, that's an excellent question, and actually one of the main aspects why this is a challenging problem. So let's go back to this 3 billion letter book. So within this information, we have all the genes that code for the proteins that you were mentioning. So in human, so to speak, like 20,000 genes. So 20,000 units of information, each coding for a protein. Then all this kind of information is the same in different cells. What changes is what genes are active in different cell types, what units are working or not. And this information is encoded in the surrounding contexts of the gene within this book. So we have regulatory elements that regulate how each gene is expressed and active in different cell types. So cells have all the machinery to read the genome, and different cells have different machinery. So they will read different parts of the genome by activating or not these regulatory elements. And that's why understanding DNA or the genome is about understanding how all these elements are placed inside the genome and how they interact with each other. But it needs to be done in a context-aware way, where in different cells, the mechanisms, the interactions will differ. And that's what makes it challenging, and why I think that you really need large-scale data, machine learning models to kind of cope with this complexity, unify all this information into some actual understanding of the code.
And maybe compared to like natural language, how far apart are these bits of the DNA that sort of are correlated with each other?
Yeah. So of course it depends on our knowledge. It's evolving over time. I think nowadays we know that at least up to 1 million nucleotides long, these interactions can occur. Of course, less frequent the longer it is, but still important. And maybe more important than that is not only that the information is encoded in a long context, but it's very sparse also. So it's not that all the 1 million nucleotides matter for this particular property that the model is predicting, but really the information is scattered across that region. It's hard to identify where the information is, how it interacts between those different regions. And maybe that's one of the main differences to other problems, where, for example, in natural language, maybe all the 1 million matters and makes sense. In the DNA, we have a lot of just maybe you could say randomly evolved sequences that don't really maybe matter, and the model needs to be able to identify what matters from what doesn't matter for a given property, and that's key compared to the other approaches.
Yeah, it sounds very challenging because you kind of need to distinguish between signal and noise, I guess, and
exactly,
really long context.
And this signal to noise also differs between different cell types, different contexts as well.
And just for context, how far away can these kind of elements that regulate what a cell is going to pick and choose out from this, how far can they be from the actual kind of region of interest, so to speak? What's the context basically that you need for it?
Yeah, so I think we are still learning what is the actual context. In the end, you could say that the 3 billion letters are the context, so that's the maximum. From the experiments that the field have pursued, we know that at least 1 million nucleotides is maybe most of the information is encoded within these 1 million nucleotides. So I would say if we as a field can develop models of 1 million contexts, we are already in a very good place. And that's what we tried also with this NTv3, push to that that range.
So if I want to really understand kind of a gene and how it's going to behave, I really need to not only look at the gene itself, but also kind of plus minus 1 million roughly.
Yeah, exactly. Yeah. But what is also interesting is that the information is very sparse. So I'm not saying that all the 1 million nucleotides are important. It's just the information is somewhere there, but it's hard now for the models to identify which regions actually matter and how they matter, how they interact with each other to regulate genes and so forth.
So to understand that a bit better, because I'm really not familiar with how biology works. So this context basically is saying, okay, if you want to express this gene in a liver cell, then you should do this, and this. But if you are in a different cell, then you maybe don't express it at all.
Exactly.
Is that correct?
Yeah. And what dictates that are regions around the gene that are active or inactive that then activate the gene itself. So you have a lot of proteins coming in and out of that locus, that region. And in different cells, you have different proteins. So they will bind to different parts of the DNA and activate different parts of it to then activate or not the gene. And sometimes not only on and off, but it's really the levels of the gene that vary between different cells. And even inside the tissue, for example, liver, you have very different cell types inside liver that all need different levels of each gene, etc. So so it's really complex in that level of regulation from the same code.
And this is not even going at the sort of epigenetic level of things. This is just purely genomics, right?
So I think epigenetic, then it's all these modifications that I'm talking about of activating or not genes. That's the epigenetic part of it. From the same sequence code. So you don't touch the sequence in the cells. What you touch are these regulation and regulatory marks that you could call epigenetic. So it's on top of the genome, all the regulation that exists. So that's this layer of regulation on top of the genome. But all that is encoded in the genome. So where to change some epigenetics or not, staying very broad, depends on the local sequence feature. So you need to understand the sequence to also understand the epigenetics of it.
Right. Super fascinating. So your latest attempt to tackle this problem with machine learning is the Nucleotide Transformer family of models. And I believe you're now at the third iteration of the models.
Exactly.
Could you maybe explain what makes this third iteration special compared to the previous ones?
Yeah. So the previous iterations were maybe a simplified version of looking at the problem, because it ignored a bit this idea of the same code being different across cells. So just wanted to look at the book by itself, finding patterns, what regions of this book matter, also across different species, different individuals. And then it every time required a second level of work to make that model usable in different applications, different cell types, etc. So what we wanted with this new iteration, it was shifting this mentality of different phases of development and usability of a model, and bring everything together. So unify all these steps, and at the same time learn how the book by itself is structured, how all the information is structured, but also all this information is then used across different cell types, across different tissues, different types of genes, different types of regulatory processes, etc. All at once with a unified training paradigm. And that we can talk about later, but given that, it also allows for generation of DNA sequences naturally having all these contexts during the first training phases. So that was the main shift. So unify different paradigms that until now were kind of kept separate, and also bring more and higher quality data, which is also key for this problem.
Okay, so and we've seen this before in other episodes, right? Like if I go back to like the previous first episode, for example, with Alex, like the paradigm of gather all the data that you can and try to model it all with one model. Then you can specify different tasks, but all from the same model. Is that more or less the direction?
So you see it across different fields, and I think we and others now at this time are bringing that idea to genomics as well. So that's the ambition now.
Nice.
So maybe I'd be quite keen to hear a little bit more about the data that you're training on, right? So this must obviously span a number of genomes, a number of species, right? How big is the data, and how do you go about kind of dealing with it, right? Managing the data itself and curating it and so on and so forth.
So I think now that there are kind of two types of data. So we have a lot of sequencing data from various species and individuals without the functional part, the gene regulation part. So that's just sequencing like genomes. So that's like these books. So we have a lot of books. So for example, in our data, we have around 8 trillion nucleotides, so more than 100,000 genomes from various organisms, etc. But that data is not annotated at the functional level. So then when we go to the functional type data, so that means that people had to do experiments in cells or in tissues to extract which genes are active, etc. So that's more costly and it's more limited in terms of data nowadays.
So annotation is kind of like highlighting pages of the book and saying, okay, this is
what regions of the book matter, and also how you can do the same experiment in different cells. So now you see, okay, this region matters in cell A and B, and this other in cell C. So that data is much more limited, and that's why people have before done pre-training only on the first type of data, so without caring about that at the beginning. Now we are at the stage that we already have enough data, but for a limited set of species. So the ones that people use the most for clinical applications, of course, human data, but then model organisms for research, plants as well for agronomics, etc. So we compiled a large data set from more than 20 species. But you see from 100,000 to 20 species, so it's a big difference. And there we are around 16,000 experiments, so 16,000 regulatory data for each nucleotide, which is already a lot, but it's not like trillions of information. So what we wanted was to don't choose one or the other, but really combine the two together. And so we did that in our pre-training strategy by combining the first type of more function agnostic data, just the book, and the second where you highlight what parts of the book matter to try to get the most out of it.
So you went for a multi-phase training approach as well?
Yeah. Like a curriculum approach where you first do this large-scale unbiased approach, and then second, you tune it to this functional data. So after you learn the global structure of the genome, now you tune it to these experimental data, these species, these tissues, these cell types, etc.
And obviously, one challenge there is this context that you require, right? So it's the kind of the actual length of the sequence that you need to feed into the model. How did you get around that?
Yeah, so to give you an idea, the latest version V2 of the Nucleotide Transformer, the maximum length that we trained on was 12,000 nucleotides, so very far from the 1 million. And now we are at the 1 million. So the main difference, and it happened with other research groups in the field, so it was also inspired by the work of others, was to move from a pure transformer type architecture, which was the core of our previous iteration, that is limited, of course, in context, to a UNet type architecture, where you still have the transformer at the middle of the architecture, but before and after you have some convolutions to downsample and upsample back the sequence context. So then you can play with the number of convolutions depending on the computing that you want. But it really allows to kind of process 1 million sequences and predict at single nucleotide level, but at its core, the transformer sees kind of more lower resolution summary information of different regions. And so you can process with the transformer 1 million long sequences, but with this trick that the transformer doesn't see 1 million tokens, but sees much fewer, for example.
Yeah, so essentially you go from like the big sequence of say 1 million nucleotide bases, you project that down to a much smaller vector.
Yeah, of like 4,000, for example.
Right. You do your
tokens.
Transformer there, and then you
upsample back. Okay. Yeah. So you kind of find a compromise between processing long sequence at single nucleotide, but using a transformer to do that core work of interactions between regions.
But inevitably, if you do this, you will lose some information. How do you deal with that when you're upsampling again, making sure that you're sort of not just inventing stuff?
So architecturally, we have, for example, skip connections that help also the flow of information. But to be fair, because like I said, DNA is not about individual letters per se, but it's about patterns. So since the beginning of machine learning in genomics, that convolutions have been very powerful for that, to extract patterns. So they are actually naturally, similar to what maybe happens in DNA and cells. So for us, it was very intuitive to actually use convolutions to do that job of downsampling the information, but keeping the patterns that matter, that are learned by the model, such that information is not lost at the transformer. Because to be fair, I don't think we really need to have every nucleotide interacting with every nucleotide. If you actually find a pattern at 100 nucleotides each, might be enough to then look at interactions between those 100 nucleotide regions themselves. So that was our intuition, and also based on research in the field.
I mean, you were also saying that the information is very sparse across the genome, right? So that's kind of a nice side effect, is that you're getting rid of that.
Yeah, you pull the information.
I guess the dead zone, essentially. Really, really interesting. And so once you have the trained model, right? So you have a model that is aware of both sequence and kind of different attributes, labels, so on and so forth. What can you actually do with it?
Cool. So I think in the paper, what we tried was to create almost a compendium of different areas of the training, but then also different types of applications. So the first thing you can do is exactly what we're talking in the beginning: predict from the genome what happens in different cells at this regulatory level. So what genes are active and etc. So that aligns with this experimental data that we used in this pre-training second phase in this case. So you can predict that. And then so you can do that for one species, but that was part in the training. You can do that for new species, for example. You can do that for new sequences. Imagine you have now variants in the sequence, for example, mutations. You can now predict how that change in the sequence can disrupt all these regulatory profiles. So you can use it for variant interpretation. We have some cases in the paper as well. And then ultimately what we did was also, the model learned a lot of information altogether. Can we use it to also improve some sequences, design sequences de novo, for example? So I always like to see this as a three goals for each model. So you want to predict for new sequences, you want to understand the impact of a variant or just really how these genes are regulated, and you want to design new sequences. So we believe with this model we could do the three, and we show in the paper examples for each. But of course we are following up with more robust studies on each and specific applications. So this was just an illustrative cases, let's say, of how you can use the model, and we open source the model for people to also use it on those applications.
So on the function prediction, are people actually using this now to inform experiments to improve the sort of the data that is available in terms of like associating function to genome?
Exactly, and we do it ourselves with customers as well. So you can, and there are two areas. You can just take the model as it is and predict within the distribution that was used in training, or you can also just take the whole model, fine tune on a different data set, a different cell type, for example, and you get top performance. And we benchmark that in the paper. So one of the main interests is using this model on their own data, their own case, and we also provide pipelines for them to do that by themselves. So that's the motivation for having a very large scale model, is that it's easy to adapt for different applications.
So you already alluded at this now with your answer that so you with this model you can perform in silico experiments and then you can compare to actual in vitro experiments.
Exactly.
So from a design perspective, how do you make sure that your in silico experiments are sort of faithful to reality?
So for example, in this case, what the use case we used was generating sequences with a given functional property. So in this case was sequences that regulate genes. So if we come back to this point of how genes are regulated. So the regions are usually called enhancers, so that's the term. And we wanted to design enhancers that activate a gene in a specific context and with specific activity levels. So you don't want to go to the lab directly until you are confident about the sequences you generate. So what we did was to have kind of in silico oracles in a way that guide you and tell you how close are you from achieving that result. So kind of orthogonal validation still in the computer in silico. Also models from other labs that were predicting the same part. So try to see if the sequence you are generating are aligned with what you want based on these oracles. So we guided the development of the generative part of the model like that. Once we were confident, we collaborated with people that are experts in these experiments. So Alex Stark in Vienna, and we tested a subset in the lab that showed the results. Recapitulated what we wanted as what we prompted the model to generate. And it's really a proof of concept for people to use for other use cases as well.
So this guiding process is not part of the training, but you use it during inference. Is that correct?
So we have a training phase where we adapt the model to a generative setup. It's a very natural follow-up from the pre-training phase because we pre-train with this masked language modeling approach. And then we use a discrete approach for generation, but that is based on masked language modeling. So it's kind of masked diffusion language model. And so what you do there is you tailor the generation to the data set and the task of interest. Because during pre-training, it's more open-ended, the learning process there. And here you really tailor it to, I want to generate enhancers in this context. So the model will see a few examples, try to generate based on the training data, and then you can extrapolate to new sequences from scratch afterwards. So you need this phase, and we provide pipelines for people to apply it as well. But so that really optimizes the performance towards the task of interest.
So then you could also, in theory, if you, for example, had experimental capabilities and you were running lots of high throughput assays, let's say, then you could, in theory, also just take the model and fine-tune it to a generative task that is completely bespoke to what you're wanting to do with it.
Exactly, yeah. That's the idea of having this foundation model. You can take it and now do a lot of different things, different contexts. That's one of the main type of things you can do with the model. We showed an example, but there are many parallels that you can follow.
And really interesting also is this idea of kind of prediction of perturbation effects, right? Can you kind of give an example of how that would be used in practice, where it might find application?
Yeah. So for example, one first aspect that I think it's very important within research is really just to understand. So maybe not in terms of practical application at this phase, but can we understand how this book works, where the information is, how it interacts, etc. And there is a lot of work over the many years trying to interpret these models. So we leverage some of those techniques like attribution methods, in silico perturbation, to take some use case and understand, okay, how is this gene regulation? Where in this 1 million long sequence is the information key for this gene to be active in cell type A and inactive in cell type B? So we did that pure of interest of understanding how biology works. And then you have the second aspect, which is, okay, if we understand our region works, we can understand the impact of perturbing that region. So we have some examples in the paper showing how you can interpret the model and identify how a variant impacts genes and all the different mechanisms that exist. We show examples for each. So I think we still need to make stronger evaluations of the capabilities of the model at this variant prediction, and now we can actually use the model there, clinical applications, etc. We are not there yet, but we show that this training paradigm helps with that, and that should be the way forward for the field.
I mean, super cool. Really interesting to hear all these applications and kind of the different use cases that you've thought about. Obviously, we've barely scratched the surface here. And I just wanted to see what you think the kind of future direction of this field is, what your work at InstaDeep and the team's work is going to look like, and kind of where you hope to take the models in the NT family and the research area stream, so on and so forth.
Yeah. So I really believe that as a field, this is the direction. So models that unify a lot of paradigms and data modalities as well. So this idea of models that go from sequence to function to understanding to generation, all with the same framework. And we hope to stay, with that type of approach, but bring more data, more modalities, and that will make a big difference, I believe, as well. So even, for example, personalized data. For now, we always train on a kind of a reference genome to functional data. Can we bring functional data from different individuals? So bring variation also at the input sequence level. So different variations of the book, for example. So to me, I think that's the direction we are taking as a field. And then second, that one aspect that we also, it's also strong in this paper is openness as well. So we as a company also really try to share with the community all these foundations. So our paper has multiple pages explaining all the details to the best of our ability for people to see how we did and build on top of that. But all the models are open sourced. We put a lot of work into very many tutorials on how to do all these aspects we touched about. Training on new data, understanding, generation, etc. All open sourced, and we hope people can build on that and bring these models to more use cases, more applications, and change in a way medicine and personalized medicine with this type of models.
That's brilliant. Thank you so much for joining us today, Bernardo.
Thanks for having me, guys. Yeah, really a pleasure.
And to any listeners who might be interested in trying out the NT family of models, we've linked in the episode description links to our GitHub and Hugging Face, where we have the models themselves and also a number of resources that will help you get started with them.
And make sure to pop by next month, where we will continue our discussion on genomics, but this time focusing on applications and real-world use cases. Bye-bye.
Hi, everyone.